Welcome everyone to our last plenary of this AIMOS conference presented by Professor Joan Leach. Joan has been based in Australia since 2005. A decade before that, she was wrapping up her PhD in Rhetoric of Science at the University of Pittsburgh. And before that, she did a double degree in English literature and biophysics from the University of Illinois. So now, here at the ANU, Joan is the director of the Australian National Centre for Public Awareness of Science, also known as CPAS, a role that she's held since 2016. CPAS was the first centre of its kind in Australia to prioritise science communication outreach, education, and inter- and transdisciplinary research. Joan's own research is based in account of knowledge production from the field of social epistemology. For 9 years, she edited the journal Social Epistemology. She's the past president of Australian Science Communicators, a founding member of the American Association for the Rhetoric of Science and Technology, and she's the past chair of the National Committee for History and Philosophy of Science at the Australian Academy of Sciences. Today, Joan will be giving us Metascience insights from the world of stem cell research. Please join me in welcoming Professor Joan Leach.
Thank you. Lovely introduction. It made me 10 years prior to that. I was like, oh god. Okay. But hopefully, yes, I will give you some Metascience insights today. I'm hopeful of that. So stem cells is what I'm talking about today. This is my sort of, I guess it's a bit coy hook, why you should care about stem cells, even if you don't care about stem cells. Look, I think we're in for a bit of a wild ride over the next few years. And so Robert F. Kennedy has sort of made a rather public play for the FDA. And just this week, he's been talking about, well, he's been talking about raw milk, he's been talking about heavy metals, he's talking chelating techniques, and he's been talking about stem cells. And he's been heavily invested personally in stem cell, in the stem cell industry in the US, private clinics. Using stem cells derived from fat for about the last 10 years. Made a lot of money. There's a lot of money to be made. So that might be one reason why you might care. So of course, in 2019, a judge ruled against this one stem cell clinic. And we were like, oh, wait a minute, how big a problem this is? Well, it's a huge problem. Okay, and it was a problem in Australia as well until 2017, the regulation changed. And a lot of these clinics were regulated. They were basically making promissory notes, as does Bobby Kennedy, about stem cells being treated for quite desperate conditions. Okay, so you've got cancer; it's no longer treatable, and so they're advertising treatments based on your own stem cells in order to resolve the cancer. Also for diseases, especially in children, it's another place that's touted. So this is quite desperate stuff. And Kennedy's wish list for the FDA is to deregulate in this area. So I loved the good news that we just had in those talks, but I think we're in for a really interesting ride, especially in this space. So that's one of the reasons why I wanted to talk to you about this project today. And Kennedy said earlier this week that he's very happy. He says, we don't need to regulate this space because there's great evidence. I have nothing to hide. What do you have to hide? And I'll come back to that question at the end. All right. So this is me talking through a project that is funded by the MRFF, Stem Cell Therapy's mission, and it's called Enabling Openness in Australian Stem Cell Research. You see the S and C in small letters, and that's because we actually hoped that it wouldn't just be about stem cell research, but we'd have something to say about all research. I think we do. We're getting there. And maybe in the conversation we have today, some of those bigger issues will come out, which I would love. So the idea, there were two big ideas behind this. One was we were going to be an interdisciplinary research cluster interested in trust in stem cell-based research and therapies, and what anchors that trust. Okay, so that's both what anchors the trust for researchers that do research in stem cells, and what anchors that trust publicly. And what's the relationship between those two things? The relationship between those two things we're still working on, but the first two, we have some ideas. Examining different understandings and the limits of openness among interested parties in Australian stem cell research. So we're talking about sharing data, sharing material, stem cell lines, which I'll talk about in a minute, collaboration, and also the kinds of communication that happen in the space. That's what we set out to study. And it's kind of interesting because we actually assumed a whole bunch of stuff when we put in our grant proposal. And boy, I don't think we had questioned those assumptions quite. And so that's kind of going to come out. It's like you put in a grant, you think you're going to study one thing, and you realize, oh, wait a minute, we got to do this first. And then we got to do this work. And then you realize you're in another parking lot somewhere because you had to back up so much in terms of your assumptions. It's been a little bit like that with this project, but I think that's good. It has been a bit like that. We thought we might be doing the kinds of studies that we just heard about this morning, the good news ones, looking into the data and that the shareability of the data and all that stuff. And then we realized, oh boy, before you can adequately do that, there were a whole set of assumptions about values and norms in this field that had kind of gone uninterrogated. And so that's kind of where we ended up doing just a real bulk of the work. We also thought we might even be doing like an Ostrom-like knowledge commons kind of study. That was so exciting to me about 4 years ago. I thought this would be excellent. This is an example of a place in science where we can see a knowledge commons in work and we can document that, and that's going to be awesome. Well, that was wrong too. And so there are all kinds of things that kind of, but I think it's been incredibly productive. And I hope we'll be able to make a bit of a contribution in this field. So this is all of us, mixture of people who work in philosophy of science, history of science, ethics, legal studies, and stem cell research. So it is truly an interdisciplinary team. It's been very, very interesting having conversations amongst the team. Again, very different assumptions about how things should go forward and the challenge of working interdisciplinary, but we're up for it. I'm very pleased here at ANU to be joined by postdoc researcher Dan Santos, who's actually running another conference in a building 2 doors down right now, and the fabulous Tommy Doe, who's actually in the room, and he just joined in the last semester. He is a second-year student here at the ANU and doing fabulous work. And I think I mentioned this not to embarrass you, Tommy, but I probably will, but to say I really think it's incredibly important that our undergraduates, from the moment they arrive at university, have some kind of touch points with these issues of metascience, because by the time you're talking to people in their postdocs, it's too late. Right, it's really, really difficult. So I'm just really thrilled that Tommy's come on board recently to help us out. All right, so that's us. So what I'm going to do, quick introduction to stem cell research. Just a few things about the science that are useful to know in a kind of historical way because they come up again and again. So if you don't know this field, this is kind of the very quick lay of the land. I'm not going to get all science communication on you and teach you science, but just a few things. Some metascience issues that have motivated us and have kind of blown up the project at certain points. Talk a little bit about my own field of science communication and what it does and doesn't bring to the party. I might be a little bit critical about my own field. That's okay. And then some remaining complexities, because this weekend, the project does its last bit of empirical data research, and that is a public deliberation on stem cell research. We're doing that in Adelaide. It's a randomized group of people, and we're going to have a whole day with them. And that's a big deal to me because that's actually when you really find out what kind of traction you're getting with certain kinds of arguments. So extremely excited about that. I'll just say a few words again. Okay, right, stem cells. Actually, we need a history, a really good history. I'd love a historian of science to pick this up because the history of stem cells tends to be told around Dolly the sheep, which is kind of like one of the least interesting things I think about the history of stem cells. But if you go all the way back, and you, Rose, you work in ecology, all the way back to Ernst Haeckel. Remember the guy, you know, phylogeny, or ontogeny recapitulates phylogeny guy. He was interested in stem cells because he thought they were kind of Ur cells, that they were going to be like the secret to understanding all life, right? And so that's how this got started. The interesting thing about that is that stem cells the study of stem cells has usually been located in research centers in science, okay? Not in clinical research centers. Now there's some exceptions there, of course, but that's interesting because a lot of the norms and values going into stem cell research we have discovered are scientific norms and values and not clinical ones. And yet, of course, the promissory note, the application of stem cell, as I mentioned with Kennedy at the beginning, are all toward the clinical side. So there's this really interesting mismatch of norm referencing that goes on in this field. So stem cells get coded as biological entities, but not so much clinical entities. Until then, they're sold out in private clinics or for very experimental treatments at the moment. So it's really kind of interesting. And you'll see that in some of the quotes. This is going to be a really languagey presentation. I'm going to give you some hopefully thick, chocolatey data of actually people talking. And you will see some of this reflected in those quotes. So until 2006, the main problem with stem cells was that they were taken as biological samples from embryos, okay? And I'm being a bit flippant here. Note that you cannot both become the embryo and end up in the Petri dish. So these embryos were destroyed. And so this was seen as the kind of big limitation to this kind of research, that in order to sample stem cells from a week-old embryo, and these stem cells would then they're pre-differentiated here, they will become all the other tissues of the body. We get them before they differentiate. That embryo has to be destroyed. And so this was a serious ethical barrier for obvious reasons. We don't want to be destroying lots of embryos to collect stem cells. In 2006, that problem was pretty much eliminated by Shinya Yamanaka, who found the way to take any kind of somatic cell. So these are not germ cells. This is your skin cell, heart cell, liver cell, mesenchymal, blood cells, whatever cell it is. Cultivate those cells in a dish. Add some secret sauce. Okay, these are those Yamanaka factors that basically have the cells revert back to what they used to be in embryonic form. Okay, so they become pluripotent. So it's kind of reversing them, right, into a state where then they can become other things. It's amazing. Really, you think about it, it's whoa. Okay, so these pluripotency factors, this induction, those lovely little triangles floating around there, that is what is heavily proprietary in this space, okay? What are the factors that cause these cells to kind of revert back to be able to be anything else? Okay, so after you revert them back, then you can induce them to become other things, right? So you might start with skin cells, but no, what I want is heart cells, okay? And so you induce them to become cardiac cells, or you induce them to become kidney-like cells, or you induce them, etc. And you will have heard of things like brain organoids. Yes, we can induce them to become cells that then get the characteristics of brain tissue. And indeed, you can, even through various modes of induction, you can grow small organoids that will actually do things like be able to have electrical waves that look a lot like certain forms of consciousness. Okay? So we generated other ethical issues, got rid of the embryo one, sort of, except guess what? You can also induce these cells to become embryos. So now we have embryoids. They're called embryoid models because we cannot, under regulation, grow them for longer than 14 days, but we produced another set of ethical issues. But very different ones, though, and worthy of a different kind of conversation. I'll come back to that. All right. So I think we're done with the science bit. We can relax. All right. So there's a whole bunch of developments here, and I'm just going to kind of run through them so that you can kind of see what kind of space we're entering, right? So these induced pluripotent stem cells, right, need to be generated from someone, right? So somebody's skin cells have to do this, right? Or an animal plant, they have stem cells too, another plant project, which is quite interesting, and I'll just refer to that one more time as I go along. So they need to be generated from someone. Okay, how are we going to do that? At the moment, most of the samples, 5 years ago, most of the samples would have been historical samples that they would have used to induce, right? Did people consent for that? Well, maybe some gave blanket medical consent, others did not. Okay, so it's a bit crazy, the consenting process around iPSCs. And if you've ever had a tumor taken from you or something like that, you probably have donated. I'm a little uncomfortable with that word now. The cells have been collected and can be regrown in some cases as a stem cell. So that's a whole section of the project that Di Nickel is working on the sort of legal ethics of this, because we have a whole bunch of historical samples that are under question. But there you go. iPSCs need to be cataloged, and of course, shareability is desirable. So, you know, the big story in the early 2000s was banking, right? Let's bank blood, let's bank tissues, and let's bank, as these cell lines became available in 2006, let's bank them. And there have been some really interesting work with biobanks. That's not what I'm going to talk about. In order to actually share the material stuff. Well, what's interesting about that is in the stem cell field, and I'll show you some evidence in a minute, people don't tend to go to banks to get their cell lines, do they? I find that surprising to anybody in the room. No, they just pick up the phone or send an email or send text or whatever and get their cell lines from other labs. Okay, so the banks have been less than successful in this area, and there's a big literature on that. What we were interested in is about the data. So, okay, banks, maybe they're not necessary, but what about registries? And so there's a big European-based registry, HIPSREG, and there's a Japanese registry, not an Australian registry. So, part of the project was going to be to see, well, which registries do Australian researchers use? Spoiler, they don't. Okay, some of them are on HIPSREG, but for the most part, not. And so there's a kind of an offshoot of this project that Christine Wells at Melbourne is running, where she's actually building the registry, which is extremely exciting. I'm going to say a little bit more about the problems she's come up with too. Okay. And finally, these material and data affordances of these pluripotent stem cells are kind of a foundational assumption. So when we started this project, everybody's like, oh yeah, they told us a story about how to get these cells, they're easy to get. And we use them, and we collaborate willingly with other labs, and everything's just this really rosy picture. And we started pushing on that picture and actually found a very different one. A picture where it looks a lot like a cabal, where you have established researchers in pretty sophisticated labs doing really, really super interesting work. But if you're a junior researcher in that lab over there, you're not going to get access. And so really interesting stuff. So in the public domain, people are starting to get really interested in what developments can we anticipate from iPSCs. So right now, you cannot take fat cells from someone and cure their cancer. Okay, sorry, Bobby Kennedy, but that's not going to happen. What you can do, though, is you can grow cells and tissues that are fantastic diagnostic tools that can replace animals. And that's, of course, what Yamanaka originally wanted to do. Remember, stem cells, biological, like the clinical stuff comes out almost outside the mainstream of the field. So that's what people are working on, a lot of diagnostics. But what other developments can we anticipate? People are reading in the news that we have organoids, we have embryoids. Are you going to be able to build a fake embryo? Can you replace my brain? And so there are all kinds of very interesting public-based questions around that, and a whole bunch of hype. How do people from whom cell collections are taken know to what they're consenting? And that's one of the things we're going to be probing next week with publics about how they feel around sort of anticipatory governance issues. Like, okay, I'll give you my skin cells for cancer research, but I would really rather you didn't build embryos with it. I could say that, right? But is that going to hold us over for another 10 years or 20 years when you can keep these cells for that long? Or my line might continue. So we've got, for those of you know, the Henrietta Lacks case, we have the potential for thousands of Henrietta Lacks cases. And so what are we going to do about that? And so what other kinds of conversations do we need to be having? So these are all the things that the developments that have happened since 2006 and really haven't been tackled to the degree that I had expected that they had kind of already been resolved. All right. So a couple of Metascience observations about stem cells, probably not a surprise to anybody here. It is a high retraction field. And in the last year, of course, there was the top retraction there from the Raffaelli one was a retraction of a paper from 2002, which has the most citations of any paper ever retracted. So, right, there's a body of knowledge there that just got, which is kind of interesting and also very difficult to deal with. Now, researchers in stem cells are very aware of this. And so we started asking them questions. Gosh, there's a lot of retractions here, and you guys share your cells and you're sharing your data. And what do you think anchors that? Like, how confident do you feel about that? Because one of the things we wanted to do was we want to be able to do the kinds of things that I saw this morning, that is look through the papers, follow things meticulously. But we thought there were a couple things that we needed to do first. And that is to find out, what do researchers think is happening in this space? This is kind of looking a little like the Wild West. So what kind of stories do they have tell about their own work such that they feel confident, right? So these are the two big stem cell registries. Something like, we have a paper on this, something like 70% of the entries are incomplete in these registries. And the skip one on top is the Japanese registry, which was really kind of state of the art when it started, and it's been wound down. So it's a kind of interesting space. So that's where we decided to go first, to kind of see what was going on here and what were the attitudes to what anchored data in this space. Would registries, for example, help? Because our colleague who works in stem cells really thought that would be an answer. We thought, okay, let's test that. All right, so we interviewed 71 people across Australia. This is a study based on Australia, and that's also interesting because I think the data is quite different than you get elsewhere. 48 academic researchers, 4 people in industry. There's some overlap there. A few clinicians, just to kind of understand what their concerns were, and patient organization representatives. Now, these are the people who, these aren't individual patients. These are advocacy groups, for example, for mitochondrial DNA, or so they're representing a kind of constituency. So we interviewed them to talk a little bit about what's going on in this space. Gosh, wouldn't an Australian stem cell registry be great? And the answer we got was no. No, thank you, Joan. It would not be great. And the fabulous Menchie, who she took the early data and published it in Stem Cell Research about Australian researchers' perceptions and experience with stem cell registration. And I'll just give you a little bit of a flavor of that, okay? And we talked to them about this sort of subspecies openness. Like, many people didn't see any benefit at all to the research community. The only reason they told us that a stem cell registry would be useful is for the public. Now, this baffles me, because what am I going to do on this website? I can't even imagine, but they seem to think it was a public, it was a public benefit, right? So, well, there's not a lot of benefit if it's not accessible to the public. Not good for, just good for us researchers. So I guess one could consider having it shared in a way that is accessible to the public, because that's where I'm going to go on Saturday morning. And then benefit the public of sharing or being open with the public is they can build public trust in a science. So I'm worried about stem cells. I better go check the registry. I hate to be, but it is a bit mad. Okay. And at the end of the day, that's how we're all funded, right? So public opinion at a really broad level is really important for funding. And it's the foundation of how the science gets done. So from that perspective, I think open sourcing is important. And I think you make a good point, because we said, you seem to be talking about the public. They're like, yeah, I'm talking, yeah, that's what it's for. Like, not my point. Okay. All right. So that was really quite interesting. So it's not for anchoring the reliability of research. It's for some kind of public trust. Okay. Wow. Another thing that stem cells reflect on is how difficult openness is within their research community. Okay. So they seem to think they can be open with the public, but certainly not with each other. So one, generosity of ideas, kind of fundamental to most of science. And the ability to share ideas and be shaped by others, fundamental to the scientific process. It kind of warms the heart. It's a difficult concept to fully enact in the competitive landscape that science operates in, but it's hard. That's what it means. And so when you say open science, well, that means, what does that mean? No idea. I think, definitely having access to reagents. We just heard that wonderful talk. Access to reagents that are developed in people's laboratories that are published, freely available reagents. That concept meanders up and down, sustaining the edifice of scientific excellence through an openness to share, obligation. So there's no Hippocratic oath on science, but this is what it would be. I was quite inspired by that. I thought I had the stuff. So that's interesting. But it seems to be two big ideas here. One is competitive to enact because it's all very competitive out there. It's really, really difficult. And second, it's a fundamental norm. Okay, more into the registry now. I think it'd be valuable if we had something like that here, like HITSREG or something like that. It would give personally me a space to disseminate the information that I have, and it's a central location. It's going to increase awareness that I have this resource, as long as it's not too burdensome on the particular researcher in terms of what they would have to do to include their cell lines on this registry. I think it'd be very, very useful. That comes up in 35 different interviews. It's just way too burdensome. And that's not going to be a surprise to anybody here, but it's one of the key things that researchers say when you ask them about things like registries or banks. Just can't do it. It takes too much time, and they go on about it. But could be useful, except now, if the registry doesn't check this information, so I give the registry the information, but who's checking, like who's policing the police, right? If I give the registry the information and they don't check this information as to how reliable it is, it then comes down to how reliable was the researcher in reporting it. Oh, meta, so meta. Or did the cell line change its characteristics over a course of time? That can happen. It's possible with any form of cell line. So to me, it's just too gray an area that in order to set up a registry, can't necessarily be reliable. The registry won't be reliable. And this came up again and again too. So other reasons why you might not like the registry? Well, you got to trust that everybody's on the same page when it comes to storing cells. That's not necessarily the case. So all of a sudden now we find out all the kinds of things that people are worried about and what's going on. It's not uncommon that you contact a lab and say, yeah, we can share the line, but then they can't find it. Or it's not what they think it is. Or they don't have the metadata and need to make it robust for whatever application that you want to use. And then finally, I have a degree of skepticism based on years, and this is interesting to me, of communicating with others and not necessarily being able to confirm what we were told about what we received. Okay, so that's really interesting to me because that really came out of these interviews, that there is a web of informal but robust communication practices within the science that are it's kind of shoring up the reliability of the data outside any kind of formal structures like a registry or bank. It isn't necessarily surprising, but here it is. Pretty amazing. And also commercialization imperatives. And I won't go through the data because this was in the ABC just a few weeks ago from a stem cell colleague working at a university in Australia, which I will not name. Probably know who it is. And in the course of this article, it was talking about issues of reliability, registries, and also publications around research in stem cells. And they just came out and said it. And I think it's just the best, well, most clear version I've seen. I'm sure we're going to get criticisms from the worldwide community. There's no publications around this, but that's just the way life is. That's because they're thinking in the old-fashioned way, right? Science is business. So openness, right, is completely opposed to this. I mean, this is very far on one end of the spectrum, but this notion that commercialization is definitely against openness. Now, we have another paper coming out where there's some really interesting interviews in the middle of our data set that actually are struggling to say things like commercialization is actually the way that openness gets expressed and having a pretty complex view, right? Those are not the majority, but there's a really interesting line of argument kind of in the middle that that's worth working with. But this notion about commercialization standing in the way of things like registries. So in both outward and inward looking ways, researchers were curious, wanted to talk and talk and talk. We've got hours and hours and hours of data because it's fun to talk about this. Very curious about how and if openness works. They're not concerned, they're not convinced that it is. And then I'll start telling you about how open lab books, they don't work either. So you just get this critique. It's like turtles all the way down. And they see this in part, at least, as a communication issue. And that's interesting. I'll say more about that in a minute. Okay. And norm conflict, right? It's just all over here. Openness is both a fundamental norm of science, it's our Hippocratic oath, and it's old-fashioned, and it's got to be getting rid of. These are both sayable views in this community. So both a norm and a practice that you can't make happen. And so that kind of informed the way in which we're thinking about our approach to openness in this project. And I really like the definition from the International Science Council on openness. I know there are many definitions that are quite good. I really like this one because what we wanted to do was take this meta-science concept of openness and then look at the practices it either enables or doesn't allow and the norms that inform it. So we can kind of step back, take that step back and understand what's going on. Because I have to think that people don't get up in the morning and think, I'm going to write a retractable paper today. There's a lot of things that go wrong, and just to simply call it fraud doesn't quite get at all the systemic issues, we know that have to kind of go wrong before something's retractable. So what are these norms, and is this norm conflict actually fueling some of these issues? Okay. Enter science communication, because we could communicate about this, right? And so as one fabulous quote, whoa. Okay. Great quote. Well, okay. So how does my field help researchers grapple with their issues in communication inside their field with publics? Not well. Okay. So when stem cell science started its sort of very fast development in 2006, from using embryos to these iPSCs, the field kind of changed. And science communication didn't change very well with it. So, here's my quote. So researchers love the idea of science communication. And why is that? Because they don't want to do it. So they want science communicators to get out there. And when they get out there, I say, well, what do you want them to say? Are you worried? What are you worried about? Are you worried about hype? Are you worried about reliability? Are you worried about the shysters out here in these private things? Are you wanting to tell people how exciting it is that we can actually make brains in a dish? Because I think that's really interesting. What is it that you would like to tell people? And they're not very clear. So I think we should invite public scrutiny. Are you mad? Okay. And we should have our best science communicators, and it's such an undersold skill set. I love that. That's good. That's good for us. Science communication. We should have our best science communicators out there and facing public scrutiny and walking them through it. And based on, okay, so I think there's a huge mismatch here in what's going on about how researchers think their field might be perceived, what publics are interested in. We are just so far away that it's very difficult even to articulate what the problem is. All right, so this is some work informed by Tommy. Tommy did a Prisma analysis for me, looking at the research in science communication. And I thought, wow. So what have my colleagues been writing about this? Like, I really want to make, I thought I knew, but let's do it properly. Let's have a look. Let's see what's going on here. Not helpful. So let me just characterize a little bit of the science communication research, because I think it opens up a really fabulous research agenda here that we could continue. So when we're thinking about science communication sort of in broadly, most of the publications are around media representations of stem cells. Now, I started with the New York Times picture, and in most of our presentations, we have some kind of nice mainstream news sort of thing. So it's not surprising if you're interested in science communication, you want to know what the media says. But that's kind of swamping the field, right? Media representations of stem cells. There's one really interesting paper. Tanya Bubella wrote a thing about the gap between public research and clinical realities and how they get represented in the media, but that's the broadest paper in that set. There's also still a whole bunch of papers about the ethical use of embryos. The interesting thing here is the Japanese literature. There's a small Japanese science communication literature. It's really, really interesting. They have a completely different thing on the ethical use of embryos. They see that the Japanese researchers are kind of heroes for taking embryos out of the equation, and also for building up cells that are going to replace animals, because that was the argument that Yamanaka gave. So they have a very celebratory discourse, and that comes out in their media. Interesting. Stem cell hype, tons of stuff, mostly critique. Too much stem cell hype, 11 times. Okay. Then we go into like the public space, right? There's 3 big publications on public attitudes to stem cells. 2 of them are international and quite interesting because those are the big Eurobarometer studies. And so they give you like a little bit of a snapshot about what public attitudes to stem cells are. So they give us a little bit of data about this kind of gap we've got between researchers and publics. Then we have a small literature on science communication techniques. Gosh, what can we do to better communicate stem cells? We could do new media arts, we could do drama documentaries, we could do blog posts. And so these are kind of really those kind of detailed things that are fabulous, but you kind of think, huh. So there needs to be some meta work there to sort of talk about the range of techniques that are being used in stem cells, in stem cell communication. I think there's a lot of work there that can be done. And then because we're like every other field, we do a lot of navel gazing. And so then we talk about our models and whether our models are good or bad. And the punchline is the deficit model of shoving information down people's throats, that's bad. And engaging with people where they start and where they're at is good, and be reflexive about your practice. 12 papers, 3 sentences. So I think that's our navel gazing kind of moment. But what's starting to emerge from our work is there's like a whole nother layer here of where internal scientific research communication meets up with public communication. So we've got one publication now out of the group about stem cells and reliable data and whether that matters to people and matters to researchers. Seems like a basic question. And how do we talk about that? So that we're starting that work. Stem cells and openness. We've got one paper now in press, right? To say, gosh, the thing is about this transformational science, right, which is the buzzword now. The thing about this transformational science is it needs an entire reliable edifice behind it. What's that? Let's discuss that, right? So we're starting to do some work there. And there's absolutely nothing on research on science communication inside stem cells. And basically that is what's running the reliability economy within stem cells, their internal communication. So I think that is where we need to go. Okay, so what's not in the literature? Well, I think the ethical issues from embryonic stem cells have so overwhelmed people that we haven't kind of switched to the new realities and the new ethical problems that the IPSCs are starting to raise. There's not a big literature there. There's stuff starting to come out in the ethics literature, but we're going to have to be able to translate that and talk about how we're going to communicate that with patient groups in particular. And that came out very much in our interviews, people feeling like they just don't know what's going on. And if you don't know what's going on, the guy saying, hey, and I can take your fat cells and cure your cancer, starts to look a lot more, realistic than it would otherwise. Attention to hype as a topic has obscured the centrality of internal communication in science. So if you ask researchers why we need science communication, they do get specific, they'll say it's to get rid of hype. Okay, well, that's fine, fair enough. But you also need a positive message, and what is that? And that has not kind of been thought through, I don't think. There's an emergence of trust discourse, and we participated a little bit about in this. So you'll hear there's a crisis of trust in Western democracies, and that crisis of trust has enveloped science. And so people don't trust institutions and people don't trust science. I hear a lot of that, and stem cells have kind of started to get caught up in that general people don't trust anything kind of thing. I think that's a worry, because I don't think we're being very, very specific when we're talking about trust, and there's a whole critique here we can give into that trust discourse. And also to think about when you're asking somebody, do you trust this? You're asking them for a yes or no answer. And what I know from my years in science communications, when you ask a public a yes or no answer, be prepared for the no. And frequently scientists are, oh, well, we didn't want you to say that. And so that trust discourse sort of pushes along those sorts of things. Also, the difficulty about talking about the public good. So I mentioned the commercialization aspect here, where people see openness and commercialization, and stem cells is just completely at odds. You can't do both. However, in some of our data, what we see is people struggle toward articulating a public good, right? Researchers want to do this. but boy, they can't talk about it. So back to that internal communication in science. I think one of the things that science communication can do for these people is to give them a narrative or help them develop that narrative about what public good in this space actually looks like. Because right now they can only say, well, one day we might have treatments. That's just not, I mean, okay. A small amount of discussion about how actual engagement happens in this field. So I think, again, this meta work that can be done about all kinds of engagement, and that's what we're trying this weekend. And I think the literature largely ignores the role of industry and commercial dynamics. So science communication frequently acts as if communication happens between audiences, not in any context of power or commercial interests. And that's frankly just crazy. All right. So a lot of complexities here. I think these are soluble, but this is where we are, and we're trying to answer some of these toward the end of the last year of this project. The anchoring assumptions of researchers that their data about cell lines is robust. They hold this. We do a good job, but it's tenuous the moment you press on it, because why won't you? A registry seems like a good idea. No, don't want to do that. And that guy over there is unreliable. That's the first thing they say. So that is a bit worrying, right? So those anchoring assumptions don't go very deep. Researchers are skeptical about registries providing that assurance. So the International Society of Stem Cell Research advises registries that registries are the best practice and a requirement of quality science. And you've seen pretty much the gist of our data. Researchers don't believe that. Evidence of complex communication networks within stem cell research is trying to shore up that reliability, is trying to backfill what the registries don't offer. And the public is still distracted by embryonic stem cells. We got other things to worry about now. So those ethical frames have been largely left unchanged, and it's really time to do some work to fix those up. Of course, researchers want science communicators to fix it. And what is the it? They want it fixed. Suspicion that the it is public scrutiny, right? They want us to fix up public scrutiny for them and get rid of the hype, and everything will be okay. I don't think my field's providing the data to help researchers understand the field or for us to really understand what publics want. So where to hear the public deliberation next week? Back to the researchers, right? I want to talk about their feelings around communication and regulation. They seem to see communication as the solution. Regulation is the thing you want to avoid. That's pretty standard. But I'm interested in probing that relationship just a little bit more. We've got an interesting subset of the data around early career researchers coming up through this Wild West system, where they feel kind of shut out about the informal networks of both data and stem cell line sharing. And so we're going to probe what it's like to be an early career researcher in this transformational research area. And then finally, this communicating of iPSCs. Look, there's exciting research happening here. I mean, the idea that we could actually, within 20 years, come up with a kidney-like device to take people off dialysis, that is worth struggling for. Okay, that is fabulous research. But there's so much that has to happen behind for that to become a credible future. So I guess where to from here? I think we need to do some work. Some of it's Metascience work, some of it's science communication work. Because, you know, Teddy Kennedy says he's got nothing to hide. Or it's Bobby Kennedy. So he's not funny. My brain. I wish he was Teddy Kennedy. Anyway, Bobby Kennedy says he's got nothing to hide. What do you got to hide? Well, I'm not sure what. I'd be very confident in what our answer would be to Bobby Kennedy today. All right. Thank you.